Whenever you get an injection or an IV drip, you have this inherent trust that it's perfectly, completely pure. But making sure that's true, it's not just about wiping things down. It's a high-stakes, microscopic battle. So what does it actually take to win that fight?
Think about it. We live in a world that is just teeming with invisible microbes. So how on earth do we manufacture medicines that are safe enough to go directly into our bloodstream? Well, the answer is an absolutely fascinating and incredibly complex system of defense.
And we really are talking about a battle here. It's a conflict being fought on a scale you can't even see. The enemy is literally everywhere, and a single tiny breach can have devastating consequences. This isn't just another manufacturing line, it's a constant state of war. So what exactly is this invisible threat? To win the battle, you have to know your enemy, right? Okay, so there are three main culprits. First, you've got your microbial contaminants. These are the living things, like bacteria and fungi. Then you have non-living particulates. Think of tiny specks of dust, glass, or even fibers. If those get injected, they can cause some serious problems like blockages or inflammation. And finally, you have endotoxins. Now these are sneaky. They're basically little fragments of dead bacteria that can cause a massive fever, a pyrogenic reaction, even after the bacteria themselves have been killed off. So when you're fighting a three-front war against an enemy you can't even see, a simple checklist just isn't going to cut it. You need a master plan. And that plan is called a contamination control strategy, or CCS. Now, the key word here is philosophy. This isn't just a set of rules you follow, it's a whole way of thinking. It's about being proactive, anticipating every possible risk, and building your defenses right from the very beginning. This CCS is built on several key pillars. It starts with the actual design of the manufacturing plant and the processes themselves. It covers the facility, the equipment, and of course, the people, how they're trained, how they're dressed. Then you've got monitoring systems, checking everything constantly. And finally, you have to prove through relentless validation that the whole system actually works. And this brings us to what is maybe the most important principle of all. You simply cannot test for sterility at the end of the line and hope for the best. As the official guidelines say, sterility has to be built in from the ground up. It's all about prevention, not just detection after the fact. So let's see how this is all built. We'll start with the environment. And you should think of a sterile manufacturing facility less like a factory and more like a fortress. The very walls of this fortress are in fact a series of increasingly clean rooms. Each room you pass through acts like another barrier, another airlock, stripping away more and more contaminants until you finally reach the inner sanctum, the place where the medicine is actually exposed to the air. These rooms are classified by grades, from D, way on the outside, to grade A at the critical core, and the rules get exponentially stricter as you move inward. Just look at that grade A zone. The air there can have no more than 3,520 tiny particles per cubic meter. To put that in perspective, the air on a typical city street has millions of particles in that same amount of space. It's an incredible difference. And even inside that super clean grade A zone, there's another layer of defense. You have technologies like isolators, which are basically completely sealed sterile chambers. Think of a high-tech glove box. Then there are RABs, or restricted access barrier systems. They're a bit more open, but they use a powerful invisible barrier of sterile air to keep the product completely separate from any people. But even the most advanced fortress in the world needs guardians. And in sterile manufacturing, the human element is both the most critical asset and the biggest potential risk. Here's the tricky part. We humans, we shed millions of particles and microbes just by breathing and moving around. So every single person who enters these clean rooms has to go through intense, continuous training to become a disciplined guardian of that sterile environment, what's known as asepsis. And the rules they live by are absolute. And I mean absolute. No makeup, no jewelry, no watches. They have to pass a special gowning qualification test every single year. And they are required to report any illness, because even something like a minor cold could compromise the entire batch. The level of personal discipline is just remarkable. The gowning process itself isn't just putting on a lab coat. It's a meticulous, multi-step ritual. Every single item is sterile. The suit, the goggles, the two layers of gloves. And each step is performed in a specific order designed to prevent any possible cross-contamination. Okay, so we have the fortress and we have the guardians. But how do we know, really know for certain that they're winning the battle? Well,
Well, you don't assume, you prove it constantly. And this is where that whole verification and validation part of the strategy comes in. It's a relentless, ongoing process of testing, sampling, and simulating to make absolutely sure that every system is performing exactly the way it was designed to. Just look at these microbial limits. They show you just how strict this whole thing is. In the critical grade A zone, the action limit for everything is no growth. Not 1 or 2 microbes, 0. Finding a single colony-forming unit, that's just one living microbe, is a major incident that triggers a full-scale investigation. The standard is, quite literally, perfection. And the ultimate test of the entire system, the fortress, the guardians, everything, is something called an aseptic process simulation, or a media fill. What they do is run the entire manufacturing process, from start to finish, but instead of using the actual medicine, they use a sterile nutrient broth, basically a perfect soup for any stray microbe to grow in. It is the ultimate dress rehearsal for sterility. And the standard for this ultimate test? It is absolute. The target is 0 growth. If just one single vial out of thousands shows any contamination, the entire simulation is considered a failure. And that failure triggers a massive investigation to find out what went wrong. And we are not talking about a small test run here. A typical media fill involves processing somewhere between 5,000 and 10,000 units. It's this huge scale that gives everyone the confidence that the process is robust enough and completely under control for a real production run. So if there's one single thing to take away from all of this, it's this. Purity is not an accident, and it's definitely not something you confirm with a quick test at the end. Sterility is meticulously designed and engineered into every single component of the process. The fortress, the guardians, and the master plan that governs it all. Which brings us to one final really interesting question. With all of this incredible technology, all this process design, and all this rigorous validation, what do you think is still the single greatest challenge to maintaining that perfect purity? Well, it's the very same element we've been talking about all along. The highly trained, incredibly disciplined, and absolutely essential human guardian, right there at the very heart of the sterile fortress.